Now we are going to read the second part of the antigen, where we will be reading the classes of antigens. So there are three different types of antigen. One is the T-dependent antigen, the other one is the T-independent antigen, and the third one is the super antigens. Now before going about them one by one, we will first look into how immune response occurs when the antigen enters in our body. So suppose that we have this antigen that has entered our body. So whenever some antigen enters the body, there is first of all APCs coming in contact with that antigen. So these APCs will engulf that antigen and will make different fragments of the antigen within them. Okay, and then what do they do is they will present those antigenic fragments over their surface over a molecule called as the MHC2 molecule. So over the MHC2 molecules, the antigenic fragments are presented by the APCs. These are our APCs. Okay. Now, whom does they produce? They present this antigens. That is, this presentation is done to the T cell. Okay, and there is a T cell receptor. To the T cell receptor, these MHC2 molecules loaded with the antigenic fragments present the antigen, and then this T cell gets activated, produce different types of cytokines that causes the activation of the B cells, and the B cells produce the antibodies. This is in short the whole of the immune response process. Now we will read the classes of antigen one by one. So first of all, coming to the T dependent antigen. So T dependent antigens are those antigens which follow all the processes. That means they will be first engulfed by the APCs, then they will be fragmented by the APCs into the different antigenic fragments. They will be then loaded over the MHC2 molecules. Then those antigenic fragments loaded over the MHC2 molecules will be presented to the T cell receptor of a T cell, more specifically the helper T cell, and then these T cells will produce different types of cytokines which will activate the B cells, and activated B cell produce the specific type of antibodies. So see here, the specific antibodies are produced by these T dependent antigens only. Okay, now this whole concept can be understood by a analogy. What is that? Suppose you are going to uh, get some documents signed from your school. Okay, so a number of students are going to get the some of the documents get signed from the school headmaster. Now you are there are different types of students in the school. So some students are good students, some are rowdy, and some are rich and rowdy students. So the good students, what do? They follow all the protocols. They go via all the channels to reach to the headmaster and get their documents signed. So these T dependent antigens are those good students only. They are following all the proper channels to reach to the headmaster. These APCs are acting as a peon here. They are presenting the documents to the clerk. Okay, that these helper T cells are clerk. So peon is presenting the documents to the clerk, that is the T helper T cells. Now these clerk, that is the helper T cells, will send some signal. That means he will analyze the documents and will send the documents to the headmaster office. That is headmaster is the B cells. Okay, so the B cells will see the documents and sign. That means there will be production of antibodies. So they are following the proper channels and getting their documents signed. Now come to the rowdy students of this school. What do? So the rowdy students are nothing but the T independent antigens. The examples are the capsule, flagella, and LPS. Please remember the examples. Okay, so those rowdy students, what do? They do not go to the either the peon or to the clerk. They directly enter into the headmaster's room and try to get their documents signed from the headmaster. So see here what they are doing. They directly bind to the immunoglobulin receptors on the B cells, and as they are going to bind to directly to the B cells, as they are directly, these antigens directly go to the bind to the B cells, the B cells get polyclonally activated and produce non-specific antibodies. So see, what happens, those when the rowdy students directly into the headmaster rooms, the headmaster gets furious over them. Okay, that means there was polyclonal activation. Furious means polyclonal activation. So once the headmaster becomes furious, that means there was polyclonal activation of the B cell. Then he scold all the students, all those rowdy students, and after scolding, then sign their documents. So scolding means non-specific antibodies are produced. Okay, so here non-specific antibodies are produced with the T-independent antigens. Okay, this is the story of the rowdy students. Now come to the rowdy plus rich students. What do they do? So rowdy plus rich students are the superantigens. Okay, they take the help of the peon in giving the bribe to the headmaster. Okay, and bypass the peon level. Okay, so they are just taking the help of the peon, that is APCs. They directly bind to the variable beta region of the T cell receptor. So see here, the super antigens are directly binding to the variable beta region of the T cell receptor. Okay, and giving bribe under the table to the clerk. So T cell receptors are the clerk. They are giving bribe to the clerk under the table with the help of the peon. And as they give bribe to the clerk, then the clerk becomes also furious and start producing different types of cytokines. Start scolding over to those rowdy and rich kids, and then send the documents to the headmaster of the school. And then headmaster, since sees all those rowdy and rich kids coming for the document verification or signing of the documents, that headmaster also gets furious. And furious means gets polyclonally activated. Those B cells once get polyclonally activated, they produce non-specific antibodies. Non-specific antibodies. So see here, super antigens also produce non-specific antibodies only. Okay, so the specific antibodies are produced only by the T dependent antigens, by all those good students only. Okay, because they are following proper routes, proper channel to reach to the to reach to the headmaster and get their documents signed. But these rowdy students and the rowdy plus rich students, they are not following the proper channels. They are following improper routes and causing the headmaster to get furious and scolding them, and thereby getting their documents signed somehow. That means there occurs non-specific antibody production, because the headmaster has become furious over them. That means B cells are polyclonally activated, so there is non-specific antibody production. Now what are the examples of the super antigens? The example is the scalded skin syndrome, that is caused by the SPEA and C toxin produced by the streptococcus, and then the toxic shock syndrome, which is caused by the toxic shock syndrome toxin type 1, which is produced by the Staph aureus. So these two are the examples of the super antigens. There are many more examples of super antigens, but you should limit and remember at least these two types of the super antigens or examples of the super antigens. So this is all about the different types of antigens, and by that analogy you can remember these different types of antigens. Remember, the T dependent antigens are the good students of your school who are following proper channels to reach to the headmaster for their documents to get signed. And then the T independent antigens are those rowdy students of your school who are following improper route. They are directly entering into the room of the headmaster and making the headmaster furious. That means polyclonally activated, and then getting their document signed. And then though there are rowdy plus rich kids, that is super antigens, who are giving under the table bribe to the peon here, under the table bribe to the peon here from sidewise, and then causing the headmaster to get furious, and then getting their documents signed. Furious means polyclonal activation of the B cell, and then non-specific antibodies production. So this is all about the different types of antigen. By the analogy you can understand it very easily. That's all for the different classes of the antigens.